Thanks very much for inviting me. Thanks everybody for coming. Now, as Monty Python said, and now for something completely different. I'm a doctor, and I work at a biotech in Holland called ProQR. How I came to this is worth a couple of minutes just as digression. So I'm a lung doctor. I worked on rare diseases like cystic fibrosis. 3 years ago, I was living in Boulder, Colorado, closing down a company there. And I have a little boy, he's 12 now. I was 9 at the time, and we have a 9-year-old boy next door, and they would all go out with Nerf rifles. Does anybody know what a Nerf rifle is? Okay, so we were completely armed. I mean, it was like the DMZ in North Korea at our cul-de-sac. And the kid next door was the worst shot I'd ever seen. And I talked to his dad and I said, does he need a new rifle or whatever? Turns out he was going blind from an inherited retinal dystrophy. It was called X-linked retinoschisis, which is a disease where you get a million little tears in your retina and eventually you lose your vision. And later I saw him practicing on the weekends with dark glasses, a white cane, and a coach to be blind. And it really moved me, and I decided to look at, I had been in an RNA company. I wanted to look and see what else was going on with RNA and vision. And anyway, I ended up at ProQR where we're working on this. This is Bella. Bella is about 2. She came to visit us at our office, and we're in Holland, and I can't explain why we have bathtubs full of balls there, because that predated her. But she's having a wonderful time playing in a bathtub full of balls. Now, you'll notice she has glasses on it, too. These aren't to help her see. Nothing in our current technologies can help her see. These are so she doesn't scratch her eyes because she doesn't blink, because she doesn't see enough to even know to do that. What I'm going to talk about today is her disease, which is Leber's congenital amaurosis type 10. The molecular pathogenesis is well understood. I'll explain it to you briefly, and how we've gone and taken this pretty dreadful disease and made an RNA-based therapeutic that appears to have promise for these kids and ultimately for many other diseases like this. Well, what we wanted to do at our company was to minimize all the variables and ask the question, even though we don't have perfect delivery systems, even though we don't understand a lot of black boxes involved in uptake and distribution, do we have drugs that are good enough now to make a difference? Some of you may remember, many of you look too young to remember, in the HIV field, we've struggled so hard for perfection that we didn't take drugs that were good enough into the clinic soon enough. And we didn't want to make that mistake. What we chose, though, to work in the eye is because you can inject oligonucleotides, even naked ones, into the vitreous of the eye, and they diffuse, they're taken up by cells, and they escape and they have activity. So there's a big black box here. The difference between doing this and injecting them, say, into the bloodstream is they don't go to the liver and kidney where they're toxic, and you don't need much. This has worked out well for spinal muscular atrophy by using the CNS as a delivery site, but the eye is even better. If you look down here, just for context, this area here, this is a cross-section of the retina, light coming in here, and this is the area where your photoreceptors are. Photoreceptors look like this, rods, cones. They have a cell body. These, if we label our drug, and put it in the vitreous, you can see even a day or two later that there's pretty broad uptake across a number of different cell types. Now, is that productive uptake? What percentage of that is active drug? It's probably a very small amount is active drug, but you can see we can put quite a bit in the eye because it's a constrained space. And I'll show you the PK for that in a minute. And so what we did was we went and picked a paradigm which is LCA10, and developed a drug, screened it through some ways I'm going to tell you about, and then actually took it into people and asked the question, how close are we to actually having a drug, and what else do we need to do to make it an even better drug? So this is the proof of concept for LCA10, but there's more than 100 diseases like this that we can treat with just this rudimentary technology, And obviously with gene therapy, gene editing, and other adjacencies, we could probably treat many hundreds or even thousands of patients. There are up to 15,000 targets in total. Many of them are quite uncommon. To get to the take-home message first, what we've done is to, in a very disciplined way, designed a progression of de-risking and confidence building that takes us from a good idea, and many of these come from academic labs, and get all the way to proof of concept in humans. And our goal for that is 3 years from beginning to end, and I'll show you the discipline involved in that. The idea behind this is we start with a certain program. Here we're starting with the CEP290 gene, which is a structural protein from the primary cilium and photoreceptors, at a time when we had great uncertainty. Do we have good enough drugs? Are they safe? Will they work? And ask the question, are they safe and are they effective? We've completed this phase in this program, and I'm going to talk to you about it. And we've progressed from uncertainty towards predictability. We're in the process of doing our second program now, which builds on the first, but is a much larger population. This is some 3,000 patients in the developed world. This is 16,000 in the developed world. And after that, we go into many parallel programs, testing different chemistries, different mechanisms of actions, etc. But in the context now of predictability, meaning we know safety, we know how to predict efficacy, and we can get down to even precision medicine at the level of a single patient with a private mutation in theory, and apply this technology platform. So our first drug, it's called Celofarson. It's also referred to in some of these earlier slides as QR110. It's for Leber's congenital amaurosis type 10. Leber was a pathologist in the 19th century. He described a lot of different causes of blindness that were inherited. Ultimately, I think 14 of them. LCA10 is the worst of the lot. These patients lose their sight in the first years of life. They can't even focus on their mama's face. There's no therapy. This particular mutation, this P699X mutation, introduces a cryptic splice site, which leads to a premature stop codon and nonsense protein. It's autosomal recessive, so the second allele has something like that or worse. It's the most common mutation in all of LCA. It's about 15% of patients, and it's about 50% of patients with LCA10. So despite that, it's about 2,000 or 3,000 patients. So it's uncommon but not rare in the context of being able to do clinical trials. We're also going to talk a little bit today about our second program, which is QR421A for Usher Syndrome. Now, Usher Syndrome is interesting because it's another protein involved in forming the primary cilium, but in this case, it's also involved in the hair cells in the ear and in developmentally. And so these kids are born very hard of hearing. Generally, a cochlear implant early in life is a big change maker for them, and they can do well. And then just to show that they really lost in the genetic lottery, around age 15, they start to get night blindness. In their 20s, they get tunnel vision. In their 30s, they go blind. And so while the hearing is largely taken care of with a device, what we're after is being able to pick these kids up at the time they're diagnosed with their hearing problem, start treatment before they ever lose their vision, and prevent them from having this double whammy of losing their sight after they've compensated for their loss of hearing. It's a little bit more common. It makes it a little more tractable a problem, but it's also a little bit harder to do studies in these patients, in large part because their defects start in the periphery of their eyes, whereas the LCA10 patients start in the center. In the center is where you read eye charts, and you'll see that in a minute. In the periphery, you have to do different outcome measures. But we think the biology is pretty much the same, and so the first program informs the second. So let me then take you through how we do this, because there's some key steps and some key learnings for how you build a platform like this. And again, the goal here is perfection is the enemy of the good. Would we want an antibody conjugated, light sensitive, super nanoparticle that you breathe through a nasal inhaler that makes your blindness go away? Yeah, for sure. Um, but, um, but that might come in 10 years. These kids need this today. Discovery. So our discovery work is pretty straightforward. In silico, we identify good targets for the technology. In this case, we want to go after this cryptic splice site and just mask it so that we restore normal splicing of exon 26 to 27 as opposed to having a stop codon before exon 27. We do that by putting a mini gene in human fibroblasts and then transfecting. Okay? So very different from the in vivo situation because we're using a transfection reagent. All we're really trying to do is drive towards activity of target and drug. And just as an example here, what you can see is, here's an experiment where we're just looking at mRNA copies per nanogram of total mRNA, RNA rather, for the normal splice, 26 to 27. And if we put in a wild type gene, this is about how many copies we would expect to see if we had 2 copies of the wild type gene. Here's where we start out with the, for instance, if you look at this one, which is 2 copies of the mutant gene, as we use a scrambled control or as we add our oligo in a dose response, you can see there's dose-dependent increase to where we're approaching the wild type level of expression, of this with the skipping. If we look at the converse outcome, which is how many still have this premature stop codon, you can see that it exactly mirrors that. We're getting rid of that transcript. And if you look at protein, full-length protein of CEP290, which is a big old gene, makes a big protein, you can see also, here's the wild-type levels of protein. We can achieve wild-type levels of protein in this system. We actually do this with about 20 candidates because while they may work with transfection, only a handful are probably going to work well enough to work with non-facilitated uptake in the eye and be safe enough. And that gets us to the next step. So 6 months, it takes us about 6 months to complete that full screen. The chemistries we use are a combination of fairly straightforward chemistries, phosphorothioate backbone, 2'-O-methyl side chains, or MOE side chains. Sometimes we use a little more exotic chemistry if we need to. The next step is the key step, which is we've generated, we've taken models from academia of growing human retinal organoids, optic cups, and applied that at an industrial scale. So essentially what we do is we take a skin biopsy from patients. We grow fibroblasts, we drive and select out induced pluripotential stem cells, iPS cells. We then treat them with a very defined cocktail of progression factors over 3 to 4 months and grow what's called an optic cup. The optic cup is exactly a correlate to the way the eye develops. You get a photoreceptor layer here. You get the retinal pigment epithelium, which actually, if this was in the eye, this would be now pushed towards the back of the eye by the vitreous, and this would then align along the top of the photoreceptors. That doesn't happen in the optic cup. But what we get is this beautiful structure here. This is a fluorescence micrograph of an optic cup in a dish that's been grown for about 3 months, and it's stained for the various human opsins, and this is all human work. I've selected out a rod with rhodopsin and then 2 different flavors of cones, all with, just to make it spectacular, we use different fluorescent wavelengths. But, it's really a tremendous thing. It's very well organized. There's even the next layer of nerve cells that grows with these things. And if you patch clamp one of these cells and flash light on it, it sees. And so you can measure function. Just to show you then the next step, now we switch. We're going to just put the drug into the vitreous with no formulation at this stage. This is Gen 1.0 chemistry. So we're looking at gymnotic uptake in the dishes. And in fact, we mirror the PK, which I'm going to show you later, which means that we put it in for a week and then take it out. This is sort of an untreated, and this is a treated organoid. You can see this looks a little more organized. What we're looking at here is the LCA10 cells don't make a primary cilium. Primary cilium is required to make this photoreceptor mechanism, the intercalated discs, the opsins, and everything that belong out there. And we're growing back some of that. That's shown here. I won't go into it in great detail. This is basically going from the premature stop codon in gray and the wild type in black in the control situation in these optic cups to where we are with our drug. And somewhere in here is the range that we dose in the clinic. So you can see we should be making over 50%. This is an experiment where we looked at our ability to just grow the outer segments, and it wasn't a great experiment, but we definitely could see that not just protein but also structure was changing. So that's at the end of 1 year. That triggers a year of spending great sums of money to do, industrial grade CMC to make the molecule, toxicology, which we only do in primates. If you do it in rodents, if you do it in rabbits, it looks very different. So you have to use primates. That gets us to the decision to develop. I just want to show you one piece of data from that. These data come from the Usher program. Look here on this side for a minute, because this is really interesting. We give a dose into the vitreous of the monkeys, and then we measure how much drug is in the vitreous with sampling. And you can see, okay, this is a log scale. This is 10 micrograms per ml. And you can see within about a week, it drops down to very low levels on this log scale, and it's pretty much cleared. We can't detect this in the bloodstream, but much of it is being cleared probably through the bloodstream. It's just protein bound and undetectable. On the other hand, if you look at the vitreous, now the scale's different. This is micrograms and this is linear. But what you see is that at two different dose levels, at that same time point where this drug is completely going away, it's accumulating in the retina. And here we're out at 120 days, and we're still almost at the same level we were at the beginning. So these drugs are retained in the retina for a very long period of time. We're in the process of establishing this further. We think it's at least 200 days, and we have data from the monkeys because this target we can measure its activity because it's skipping exon 13, that it is bioavailable out to 200 days as well. So very fortunate. It's probably a very small percentage of the total load. These drugs have to work on pre-mRNA in the nucleus, and they're probably in an endosomal compartment that's in some sort of equilibrium, but it may be 1,000,000 to 1 gradient, but there's still enough drug getting out. And one of the things we worry about is safety. Indeed, what we found in the human studies is if you give enough of this drug, you can start to see off-target oligo-based toxicities like lens cloudiness and even some retinal findings, but that's a lot more drug than you need to actually make the patient better. So here we are at the end of year 2 and ready to go into humans. We don't go into normal healthy volunteers, we go straight into patients. The trial we did last year then was to take adult patients and give them a dose that we predicted from the model would be efficacious, and then we could escalate the dose higher if needed. And then once we did the adults, we also went into kids as low as age 8. We haven't gone lower than age 8 for reasons I won't go into here. Now, the first translational experiment, and this is all about translational science now, I want to make this point. So here's a cartoon of your retina. Here are the cells we're after in the middle here, the cones and rods. And this is what our organoid looks like, right? But we can take a picture in the patients using something called oculocoherence tomography, OCT. And what we're looking at here, this white line here is the border between the outer segment and the nuclei of these. So it's between these nuclei and these outer segments. This produces a white line. And this white line is absent in patients without outer segments. So here's an example from the trial we ran. Again, here's a normal retina with this bright line between the nucleus and the photoreceptor outer segment. Here's what an LCA10 retina looks like. You can see this bright band here, which is back in here. This other one's kind of missing, and this is all blood vessels and stuff up here. So rather nerves and things up here, blood vessels back here. Here's a patient, though. She came into the study. She was 2200 vision, so just at the bottom of the eye chart. Here's her baseline scan, again, just the bright white line. Month 1, you can start to see this boundary appear. Month 2, by month 3, she's got a very beautiful EZ line or boundary between inner and outer segment. Only in the macula, because these patients have lost all of their rods irrevocably, but they maintain cell bodies in the fovea where the cones are. So sure enough, she grew them back, and that's the correlate from what we saw in the optic cups. Well, the next step is we can do what I would refer to as a pharmacodynamic endpoint. It doesn't tell you are they going to be able to read an eye chart again, but it does tell you did we engage the target and did it work. This is called FST. Have you ever had a hearing test where you raise your finger or press a button when you hear a tone? Same thing. You put a big hood on the patients, they get a red light or a blue light flashed, they press a button. And you can see these are the untreated eyes, blue and red, they can barely see. This is after 3 months of treatment with the drug in a group of 8 patients at 3 months. And you can see almost a log improvement in sensitivity to light in both wavelengths. Here's the line charts for that out to 6 months from this interim analysis, and you can see that this benefit in both wavelengths is progressive and well maintained for the full period of observation. So this is really dramatic, but it does tell you only that somewhere in their retina they have some photoreceptors that are working, but it doesn't tell you how important is that. And that comes from this. This is visual function now using eye charts. This is the eye chart that you guys probably look at when you get glasses. This is the eye chart that people look at when they're like Bella. And so you can see this size letter is about this size letter. That's about 2200. And they just have to tell you this is an E that's sleepy, or this is an E that's backwards. And so it's suitable for younger children. Here's an example of the second patient in the trial. He was 41 years old. He had been pretty much dysfunctional from his teenage years on. He was seen at one of our 3 study sites in the US in this case. He was light perception only, meaning if you flashed a bright light on him, he knew he was being flashed by a light, but he couldn't see hand motion, couldn't count fingers, couldn't read on the eye chart. 1 month in, he was the same. 2 months in, he had already improved by log orders to the point while he was still legally blind, he was at 2400 from like 22,000 probably down there. His untreated eye looked like this. He's a remarkable responder. He's interesting because his second allele is a hypomorph, which probably preserves cell bodies longer. But here's what the average group did. Here's the mean and the median. This is this 3-month response cut we did. 5 out of 8 treated eyes had clinically meaningful benefit. The red points are the eyes that were treated, and you can see the distribution versus their untreated eyes. Pretty clear there's a group shift. Even if we take that remarkable responder who's way up here out, we still have a median change that's greater than the 3 lines of vision, so 15 letters of improvement. Here's what that looks like over time. So again, it's well maintained over time. It happens pretty early as well. The final roll-up from that study basically is everything did get better. Visual acuity improved. Now the threshold is 0.3 logmar of improvement, negative being improvement. The group was 0.67. The untreated eyes didn't change. We also put them in a mobility course, like an obstacle course. They did get better. This was the FST I showed you. We also looked at their eye stability, and sure enough, they stopped having this sort of saccades in their vision where their eyes would move around, and they were able to focus a little bit better. Now, I will say that these are the worst of the worst patients because they went into a phase 1 trial with no known benefit. 5 of the patients out of 11 that were enrolled had no vision at all except for light, and they were all older. So as we move this drug forward and get into younger patients with healthier retinas earlier in life, we're we're likely to see even more improvement in the things that are meaningful. So very encouraging. We do have a large pipeline built on those successes, and we're going to be progressively building on that. We're enrolling a pivotal or trial for Celofarson now to get it registered. We're in the process of doing that same kind of a program for Usher Syndrome, and then we have a number of other programs to follow. Thank you very much.
Thank you. That's very exciting work. I was curious what dosing regimen you would end up planning to use for the clinic, and for the monkey trials, were you doing that with fluorescence tracking, or do you expect that the modified RNAs are actually chemically intact at those later timelines?
Okay, so the questions were, first of all, what do we think the dosing regimen's going to be? We think that for the molecule we're using, LCA10, it'll be twice a year. There'll be a loading dose and then half that dose, probably given every 6 months. That's based on that half-life we observed in the monkey study, but we're in the process of completing those studies for LCA10 a little bit more detailed to be sure. We actually sacrifice the monkeys and do, sort of fluorescent probe type measurements to quantify the amount of drug specifically in there. We don't conjugate. We did some conjugation early, but conjugation can affect the kinetics, where the drug goes, how it gets released and stuff. So these monkey studies, we want to have just the authentic drug.
One question. So you were talking about the need to screen for the right sequence. What is the what are the properties that make one good versus bad?
Well, so I like to tell people I'm just a country doctor, so my property is that it works. But if I put on my science hat for a minute, we're trying to be really pragmatic here. We're building the rules and the toolkit, but we don't have a priori rules in terms of activity. We have a lot of a priori rules for safety. We don't want to induce, say, TLR-mediated innate immunity. We make sure that they're long enough to not have off-target binding sites in the genome, things like that. So those rules take place. They get screened out early on in the process. When we get to the optic cups, the organoids, then what we're looking for there is, in the aggregate, that we can give an amount of drug that has a wide margin to the predicted off-target safety that we might see, and we're learning more about that as we go, and then has an outcome that leads to either structural or functional changes in the model that restores function to those photoreceptors. So our first two programs are ciliopathies, which is not an accident. It means that we're taking a paradigm that's very easy to see in the optic cups so that we didn't have to develop a very rigorous assay. We're in the process now of validating several integrated outcome measures, either fluorescent measures of membrane depolarization and calcium transients or direct electrophysiological measurements, so that we can look at diseases like our third target, which is a dominant negative form of retinitis pigmentosum, where they have the full photoreceptor. It's just the rhodopsin is a dominant negative, and we need to suppress that with a high affinity allele specific knockdown, and then measure, when we flash a light on the optic cup, does it now see light better? So it's very pragmatic. At this point, we'll have a set of rules that develop over time, and then someday we'll be able to figure out what it is about the chemistry and the molecules that drives them to have high activity.
Thank you. Okay.
Okay, thanks.
All right, great. Thank you.